after the inaugural session in the morning. Now we are in the session, technical session one, uh, day one in Water for All, Water, Food, Energy Nexus. The design of this session would be that first we have a thematic presentation on Water, Food, Energy Nexus, followed by the research papers we have received. For each research paper, we would be have 10 minutes for the presentation, and after that, 5 minutes for the question answers. So I would request all the participants to please mute yourself, and only you can unmute yourself while or when you have any questions, you can raise hand and then unmute yourself. So first, I would like to introduce Mr. Fareed Alam Saib, who is the Director Program for the Asia Foundation, and he is going to present a thematic presentation on water, food, energy nexus. Over to Fareed Saab.
Bismillahirrahmanirrahim. Thank you, Faizan Saab. Um, I welcome you all this afternoon for a thematic session on the water, energy, and food nexus. I will be just presenting the links between the water, energy, and food nexus. And that link has been informed by our consultation at the provincial levels and at the federal level. We had a consultation in each province. We also reviewed the water policy, the national water policy, the food security policy, the national climate change policy, and the energy policy. So I will be just presenting what we have found in those policies, but my presentation is based on 4 angles. The first angle, I'm just looking, the nexus, with an angle of coordination between the departments and between the ministries. Then I will be also looking, all policies from a gender link. I will be also looking all policies from the inclusion link. So I'm just starting the presentation now. Can I share my presentation because, I'm disabled now, right now, the screen sharing has been disabled. Can you enable me to share the screen?
Yes, sir. Fareed Saab, now you can check. Is it okay?
Yes. Can you see it?
Yes.
Okay.
Yes, sir, it is visible. Please.
Okay, thank you. Thank you very much. So my presentation is, if you see from the title page, the water crisis and the nexus, what it means for Pakistan, like what it means for Pakistan, what we mean by the nexus, and what it means for Pakistan from different angles, from the economic angle, from social angle, and from the political. If you see, we in the morning we had in our opening session we had a discussion on the importance of water. So this slide is just to give a reflection of how the numbers matter for us, like the water use. We are the world's 4th highest water usage economy, among the world 36 most water stressed countries. If you look into the water availability, so the water availability per capita in this country dropped from 5,600 cubic meters to below 1,100 cubic meters. This statistic is from 2010. And if we continue with the same pace, so by 2025, we will run out of water. We will be below 500 cubic meters. Agriculture, 93% of our fresh water resources is used in agriculture, with 2/3 of irrigation water wasted due to inefficiency and system leakages. Although we have the the world's largest canal irrigation system. But if you look into the efficiency of that system, that is extremely inefficient system because we haven't put a lot of work in that system to put it in track. If you look into the energy, 30% of Pakistan's energy resources come from water through hydro energy development. And the key factor is the population. As I said, if we continue with the same pace, if our population grow with the same rate, so we will be run out of water by 2025. So increased population has a large demand for water, food, and energy without adequate supply of resources. What is that interlinkages between the water, food, and energy? We all know that we use water for irrigation, we use water for food consumption, production, we use water for virtual water along the value chain. While that has a linkage with the food, with the energy, we use water for energy production and we use water for the food production, and we use energy for the water. So that has a linkage, but the climate change has an impact on all the key components, the water, energy, and food. I would just give you a reflection of the global warming on agriculture. Like, as I said, due to the climate change impact, we are losing many fronts. So if you look into the global warming effects on our agriculture, the loss of biodiversity and fragile environments, tropical forest, loss of fertile coastal lands caused by rising sea levels, more unpredictable farming condition in tropical areas, dramatic changes in distribution and quantities of fish and seafoods, increase in incidence of pests and vector-borne diseases, and longer growing seasons in cool areas. So this is like, the impact of climate change on our agriculture. I'm just coming to the Nexus framework. As I mentioned in the beginning, we had a consultation at the provincial level, at the federal level. We reviewed the policies, the water, energy, and food security policies, the climate change strategies. And we developed a framework. And I'm just giving a reflection of that framework, what we have found in that framework. And these are just some of the recommendations from the framework, which I'm just sharing with all of you. Need to conduct research on water availability in the next 10 to 20 years in various agroecological zones. We have policies, the agriculture policy, the water policy, the energy and climate change strategies, but that needs to be aligned with the expected water availability. Effective programs are required on water management for optimum use of available water resources. And the problem of polluted, irrigated, and domestic water due to drainage of untreated industrial and household water into rivers, streams, and canals should be addressed in the policy. If you look into the energy, we have found some of the areas which needs to be addressed. Initiate and support programs for energy production from agro-resources based on past learnings and the latest technology developments. Need to start renewable energy, solar, wind initiative for agriculture production. Encourage joint projects of universities and private sector innovation for low-cost wind energy technology. And important to check excessive water extraction due to cheaper solar energy. Food and agriculture policy reviews. We have found that agriculture policy need inclusion of on-farm energy and programs for alternate and renewable energy. And important to promote research-based and tested water efficient crop production system and transit by subsidy programs toward climate resilient sustainable agriculture to move away from greenhouse gases intensive production system, and addressing salinity problems is crucial in agriculture policies. From the climate change strategy, we found that climate change adaptation and mitigation specific programs to be incorporated in agriculture, water, and energy policies. The management department should integrate various facets of climate change, heat waves, drought, floods, excessive rains, hailstorms, etc. Universities to integrate climate change in the curriculum of all departments, like agronomy, plant breeding, agriculture, etc. Private sector to be encouraged for developing climate-friendly technology and products. As I said, we looked this from 4 angles. The first angle was the gender. Women's role in agriculture increasing due to men's out-migration owing to climate change and economic push factors. We have not addressed this as a key component in our agriculture policies, that how we need to mainstream gender in the agriculture policy. Women-centric agriculture program needed with induction of women staff and use of technology. Important to develop energy program based on women's specific needs, such as cooking, water collection, toilets, hygiene, fuel, wood collection, etc. And renewable energy for women economic empowerment, learning African experience of Solar Sisters, UN acclaimed initiative that engaged women in local business of renewable energy. So the overall purpose here is to mainstream women because they comprise 50% of our population. So we need to mainstream women in all policies and develop projects and programs to bring them in productive activities. The second most important component, which we looked in the review of all these policies and in our consultation, that was the institutional coordination. We found that we need to develop nexus-based coordination mechanism with relevant departments at intra and interprovincial and federal level, because most of the ministries and departments at the provincial level, they don't share information with each other. Like look into the agriculture and the irrigation department or other departments, energy department. So we need to promote that coordination. Each ministry and department can have a Nexus committee with membership from other Nexus departments. That's our recommendation, that each ministry and department at the provincial level, they should develop a Nexus committee to coordinate projects, to develop even, for the annual budget, that needs to be developed through a proper coordination. It shouldn't be, done in isolation. And in addition to agriculture, water, energy, and climate change department, important to include supporting departments such as SAS program, Higher Education Commission, PCRWR, Planning and Development, Rural Development Department at the provincial level. That needs to be, to be the part of this whole Nexus program, because in one way or the other way, they are the part of this this whole Nexus. The third important thing which we looked in the consultation was the stakeholder engagement. And we found that the system needed for organized and regular engagement with stakeholders such as women, men, farmers, NGO, private sector related to water, energy, agriculture sector. Examples, local level prioritization matrix, Alliance for Change Balochistan. Important to development international partners with FAO, FAO, that needs to be, it's critically important to develop partnership with international organizations, because international cooperation is critically important for all these three components in the Nexus to be developed. And the fourth important thing which we looked in the in our consultation is the research and data. And that's something we discussed. We are lacking of organized research and data information. Existing research on water efficient crop production methods should be integrated into agriculture extension department. Like agriculture extension department, and they are working in isolation. There are so many extension workers, but we are not properly utilizing those extension workers for the dissemination purpose. Which crops can be more adaptable against climate change, excessive heat, changing rain patterns, drought, etc. in various agroclimatic zones, emerging disease and pest attacks in livestock agriculture due to climate change. These are the key areas we need to look into, because from every angle, this is critically important for our production purpose, for our economy purpose, and socioeconomic impact of water stress, excessive heat, changing crop rotation on women, men, youth, and how to design a transition. Because of the climate change, we'll see many changes, and we need to be prepared for that transition. So that needs to be looked from different angles. I'm thankful to all of you, and will be happy to address any question if you have. Thank you very much, and over to Faizan sir.
Thank you, Farid Saab, for a very nice and comprehensive presentation. You have covered almost all things which is relevant with the water, food, energy nexus. So, are there any questions from Farid Saab regarding this particular presentation? I believe there is no question because you have presented so nicely. So, anyhow, you can ask Farid Saab maybe at later stage. So now we move to our formal presentations, which we receive from various university. So first of all, I would like to invite Muhammad Asif Jokyo Saab from Mehran University of Engineering and Technology, who is going to give presentation on treatment of distillery effluent using membrane bioreactors and investigate microbial community using 16S gene metagenomics. Muhammad Asif Jokyo Saab. Please share your presentation. You have 10 minutes for the presentation, then we have 5 minutes for the question answer. Kindly unmute yourself. First, introduce yourself and then start your presentation, please. Go ahead.
Rahim. Assalamu alaikum viewers and listeners. My name is Asif. I am belongs to US Pakistan Center for Advanced Study in Water, Mehran University Engineering Technology, Jamshoro. Today I'm going to present my research paper. The topic is a treatment of distillery effluent using membrane bioreactor and investigate the microbial community using 16S gene metagenomics.
May we have your full screen, please?
Okay, sir. Contents of my study. Sir, will I start, sir?
Yes, please go ahead.
Okay. Contents of my study: introduction, objective of research study, methodology, results and discussion, conclusion, references. The introduction: distillery wastewater is a waste product released after the distillation of ethyl alcohol from molasses. Distillery industries are the wastewater called spent wash. Only few industries are treating their wastewater effluents, but their treatment methods are not good due to the removal of nutrients. In Pakistan, large volume of wastewater effluent from the distilleries and sugar industries is directly discharged into canal, causing environmental pollutions and are also difficult for the aquatic life. The generated waste effluent from the distillery industries containing high biological oxygen demand. It contains 50 to 55 milligram per liter and COD chemical oxygen demand up to 90,000 and total dissolved solids TDS up to 1 lakh milligram per liter. Distillery wastewater treatment methods. There are number of ways to treat the distillery wastewater, such as physical method, chemical and biological method before its disposal. There are many traditional methods such as physico-chemical methods such as coagulation, flocculation, filtration are applied for the treatment of sugar distilled effluents, but they are quite expensive because they are using high amount of chemicals like aluminum sulfate, potassium chloride. They are very expensive. Alternatively, the membrane bioreactor technology is emerged. Membrane bioreactor for the wastewater treatment play a vital role in activated sludge and membrane filtration for the biological mass retention and high organic load rate. These practices can be applied for the both industrial and distillery wastewater treatment because it is a membrane separation process technology. Role of microorganism, bacteria especially in wastewater treatment. In recent studies, biological wastewater treatment using microorganism has attracted the attention of researchers all over the world. It means microbial degradation and decolorization of distilled effluent have been found as a cost-effective using microorganism, an environmental-friendly alternative to physicochemical method. Here are some examples to degrade microbial contamination and organic pollutants. A wide variety of bacterial species like Pseudomonas aeruginosa, Pseudomonas putida, Lactobacillus, and Plantrum, Ronella, Stella, and Perivotola have been reported for their activity in the degradation of organic pollutants from the distillery effluents. After that, metagenomics and bacterial identification. We know that more than 50% of bacteria cannot be cultured, so they cannot be identified by the culture-based method. Ultimately, metagenomics emerged. Why metagenomic? Metagenomic is a study of genetic material or DNA which is recovered directly from the environmental sample. It's a technique, it is a molecular technique to analyze the DNA to contain a simple a sequencing run. There are two types of metagenomics. One is shotgun and other is marker gene based metagenomics. In shotgun metagenomics, we know that all the genes are sequenced in a single run, but it's quite very expensive as compared to marker gene based metagenomics or 16S gene metagenomics. Ribosomal RNA, the universal phylogenetic marker. Ribosomal RNA are present in all living organisms. Ribosomal RNA play a critical role in protein translation. RNA are relatively conserved and relatively acquired horizontally. More conserved regions are useful for the treatment for the determining the higher ranking taxa because we know that 16S gene is present in all the bacteria because ribosomes can't translate mRNA without the presence of 16S gene. That's why this gene covered all the bacteria in a single gene. Those 16S gene are mostly commonly used. In this study, we are using the region V3 and V4 region selected for the target area for the sequencing. Significance of microbiome studies. Microbiome increase the bioreactor efficiency. It also increase the metabolic potential of bacterial composition and structure. It is possible that manipulation of these communities could be used in wastewater treatment process. The study suggested possible increase in commercial value of the microbiome that are present in the wastewater because we know that the microorganisms are bulk quantity present in the wastewater and any wastewater. So the objective of my research work to evaluate the microbial community structure in membrane bioreactor using 16S RNA gene-based metagenomics technique. Methodology of my study: initially setup and maintenance of MBR. The membrane bioreactor was installed at US Pakistan Center of Advanced Study in Water Lab. MBR was divided into two stages. One is initial stage and second one is a final stage of MBR. In initial stage, 85% of synthetic wastewater with 15% only distillery waste with the pH adjusted with the 7.4 to 8.5 by buffer solution. After that, taking the sample were taken after the 10 days of operation. One sample was collected from the initial stage and sample were centrifuged at 1000 RPM for the 5 minutes and filtered through 0.45 micrometer membrane filter. And sample was stored at 4 centigrade for further process. And same for the final stage of MBR. And that time, 50% distillery wastewater and 50% synthetic wastewater. The pH was 7.4 to 8.5. But the operation was sample were taken after the 100 days of operation. The sample were collected from the final stage of MBR. Sample were centrifuged at 1000 RPM and 5 minutes and filtered through the 0.45 micrometer. DNA extraction and sequencing. After the collecting the sample were used for the DNA extraction using gene jet purification kit. The DNA was extracted after then sent back to the Macrogen company Korea for the amplicon library preparation 16S gene sequencing by Illumina Y6 sequencer. After that, the raw data were obtained from the Macrogen company Korea. Quantitative insight into the microbial ecology to CHIME2 was used for the quality control analysis, quality filtration, taxonomic characterization. After that, statistical analysis of gene level obtained. Results. The performance of the MBR for COD removal was found above 98% during the startup of the reactor at OLR rate, organic load rate 4.2 gram per liter and 15% DSW. After the initial stage, the declined COD removal efficiency. It reached back to the 90% because it contained high load rate in the final stage. Then the COD removal back to the 90%. Then the reactor was shifted to 50% DSW, and the reactor was further operated for the 65 days with the average COD maintained. Removal efficiency 74% at the OLR rate. Organic load rate is 16.4 gram per liter per day. Table 1 number shows quality analysis of sludge sample using CHIME2. CHIME2 is
Asif Saab, you
a
have 2 minutes, please. You have
okay,
2 minutes.
Sludge two samples were two samples. Input reads are 305 and 321,285 in final stage. It contained total reads were 627 to 228. In CHIME analysis, relative abundance of major phylum in all the sludge sample. It was observed in the initial stage, the protobacteria were found 64% as compared to the final stage, 33% protobacteria and bacterial rates, 44% was observed. In CHIME analysis, relative abundance at class level in all sludge sample, it was observed that 32% was alpha protobacteria as compared to 10% in final stage, and also the alpha protobacteria 10%. In CHIME analysis, bacterial abundance at genus level, it was found unidentified or unknown genus level was found, but high abundance alkaline initial level and final stage Ronella, Holoferella, Colostrum was found in the final stage. Discussion in this study, 16S gene metagenomic sequencing approach for the identification of bacteria was evaluated on the distilled wastewater sample. Bacterium phyla with the relative abundance, including Protobacteria, Bacteroides, Firmicutes, these phyla identify the other researchers. Bacteroides found high abundance in the final state. It is a capacity to convert organic carbon to carbon dioxide through aerobic oxidation. Clostridium was reported as a pathogen agent in wastewater samples. Conclusion: COD removal was found to be 98% with the startup of the reactor. The reactor was stabilized at distilled wastewater. The COD removal was maintained up to 74% at 16.4 milligram per gram per liter per day. In 16 gene metagenomics were revealed that initial and final stage sample were compositely different. Many species were found like Holoferra, Stella, Ronella. These have the reported to have a capability to remove the phosphorus nitrification, denitrification, sulfide reduction, and organic removal. And one of the gene, Prevotola, is considered genus is a fermentative acetogenic bacteria found in final stage. Acknowledgement: Korean Institute of Science and Technology project is highly acknowledged for the funding the project. US Pakistan Center for Advanced Study in Water, Marine Exchange and Technology is acknowledged for the providing the basic lab facilities. References, more references. Thank you.
Thank you, Asif Saab, for a very impressive presentation. If we have some questions regarding this presentation, do we have a question? I believe no question. Thank you very much, Asif Saab, for your great work. Thank you.
Sir, I have a question.
Okay, go ahead, Barira.
I just have this one question that I have read in literature that effluent from distilleries already contains some bacteria. So the quality of the wastewater is already better. Have you found this same phenomena in your research or not?
Yes, of course. I have found many pathogenic bacteria are found like Clostridium and like very impressive bacteria like Ronella and Pseudomonas. These are the very important bacteria which are play an important role in the distilled effluents. I have found in the literature.
Thank you.
Thank you, Asif
Thank you,
Saab.
ma'am. Thank you.
Now I invite second speaker, Mr. Zubair Ahmed, who is going to present treatment and color removal of distillery wastewater using membrane bioreactor. Over to Zubair Saab. Do we have Zubair Saab?
Yes, sir. It's not Zubair Saab, sir. It's Bahadur Ali.
Okay, no issue.
Yes, sir.
Please share your presentation. You have 10 minutes for the presentation.
Assalamualaikum. I'm presenting my research paper here. The research topic is development of calculation and absorption system for biological treated distillery effluent spent wash. My name is Bahadur Ali, and my supervisor is Ma'am Mariam and Sir Zubair. My table contents are introduction, problem statement, objectives, methodology, scope of study, results, conclusion, references. Introduction. The sugar industry is the biggest agro-based industry in Pakistan with growth rate of 3 and 0.7 respectively. Moreover, it is produced by product spent wash. Spent wash is a dense brown watery material that comprises of BOD 45,000 to 55,000 milligram per liter and COD 90,000 to 110,000 milligram per liter dissolved solids and salts, and only few industries are treating their wastewater, but their treatment techniques not suitable for eliminating toxins compounds according to national environment. So many technologies are being used to treat distillery spent wash, but here the biological treatment for distillery spent wash with membrane bioreactor treated with MBR spent wash, but still have dark brown color with 30% of COD and has been removed with the biological treatment of MBR. Moreover, the spent wash contains high levels of chemical oxygen demand and BOD, low pH total dissolved solids. Untreated disposal of effluent into water streams may create the color of the receiving stream and the dissolved oxygen of depleting, creating a potential threat to the aquatic organism and the microbiome. Conventional biological treatments involving aerobic digestion, biomethanic, not much effective towards treating these high reactant. Therefore, other probable substitutes have been tried for degrading the wastewater color. Physiochemical treating methods involving electrocoagulation, adsorption, coagulation, and flocculation, ozonation, evaporation, and so on. So adsorption is one of the foremost physiochemical treatment method to get rid of contaminants and color from the spent wash has been employed. In spite of the adsorbent employed, the degradation process will be influenced by the factors such as initial concentration of the effluent. So my problem statement is that the COD and the color of distillery effluent spent wash results in environmental issues with discharge without any treatment into the water resources. Even after treatment from the membrane bioreactor, the spent wash effluent has a remaining COD of 30% and a dark brown color, which threaten the aquatic life. Research objectives is COD and color removal of MBR treated distillery effluent spent wash through coagulant aluminum sulfate and synthesis of activated carbon granular and activated carbon powdered moringa oleifera seeds. Use them as adsorbent to remove residue COD and color. Scope of study. This adsorption and coagulation system can be used for the removal of residual COD from spent wash effluent, which can't be removed through treatment units. Color and removal from spent wash effluent will streams from the contamination and to protect aquatic life. Material and methodology. I have carried out the test on jar test equipment. Jar test is a pilot scale test used for wastewater process using chemicals. The purpose of coagulation, flocculation to remove suspended matter, solubility, color, and so on. I used the 4% aluminum sulfate solution. Dosing was 0.2 to 6 ml at the concentration of alum 0.8 milligram to 24 milligram per liter, time of 64 minutes. And the optimized dose was the 16 milligram per liter, to where I achieve the desirable results. And I done the calculation of MBR treated spent wash after treatment which are color, oxygen demand, COD, pH, BOD, total solids, nitrogen, total phosphorus, total suspended solids, voltage suspended solids were according to alpha method 2017. Furthermore, the characterization of Moringa oleifera seeds, granule activated carbon and the powder activated carbon will be analyzed using SEM and PET test. These are the results where I done tests on jar test equipment through coagulant aluminum sulfate. I removed around 65 to 70% of COD on the optimum dose of 5.5 milligram per liter, and the color removal is also removed to that dose 6 milligram per liter. So conclusion, the COD and color removal were observed at optimum coagulant dose aluminum sulfate 5 milligram per liter, shown better removal percentage throughout the study period. pH was monitored during jar test analysis. Furthermore, this study will be optimized as different materials as Moringa oleifera seeds, GAC, and PEC. These are the references. Thank you.
Thank you very much, Bahadur Saab, for finishing your presentation in time. Do we have only one question for this presentation?
Yes, sir. I have a question. May I ask it? So, Mr. Bahadur, you have you have showed slide number 12 and 13, in which you can see that COD and color removal. The graph is not uniform. Why is that so? The next one.
This is because of equivalent dosage. It does not give the accurate results in all doses. It also creates you can say suspension during dosage. So that's why the graph is not linear.
Okay, thank you, Bahadur Saab. Now we move to our third presentation. I invite Ms. Sanam Bhatti, who is going to present to investigate antimicrobial resistant profile in drinking water of Mehran University of Engineering and Technology. Over to Sanam. Sanam, are you there? Sanam, are you there?
Hello?
Yes, Sanam, please share your presentation, but your voice is... Please share your presentation. Yes, please go ahead. We can see you.
Everyone see this presentation?
No, you have not yet shared your screen. You have to be quick because we have a lot of presentations today.
Yeah, sir, just a minute.
Now we can see your presentation, but please make it full screen.
Yeah.
Yes, it's okay. Please go ahead. You have 10 minutes.
Okay. Assalamualaikum everyone. This is me, Sanam Bhatti. I'm glad to present here my presentation. So I have shared my presentation with everyone. So my presentation topic is to investigate antimicrobial resistant profile drinking water of Mehran University of Engineering and Technology, Jamshoro. This is me, Sanam Bhatti, and two of my co-authors. One of this Professor Dr. Rasul Baksh Meher, and second is Mr. Junaid Ahmad Khori. This is our table of context that I will cover throughout my presentation. Very first, I will proceed with the overview. So overview is that in pathogenic bacteria, antimicrobial resistance is a significant public health threat. Various factors contribute to the spread of antimicrobial resistance in the environment. So this study was designed to measure the antimicrobial resistant bacteria in water distribution network of Mehran University main treatment plant. Introduction. Antibiotics are used to treat infection diseases. However, due to increased consumption of misuse of antibiotic, surface water is being contaminated. So the antibiotic resistant bacteria are spreading in environmental media such as natural river, hospital effluent, sewage treatment plants, and many others. So the today clinically importance are bacterial characterization, not only a single drug, but also multi-drug resistant, that also called MDR, so multi-drug resistance. This is one of the big issues. This is a literature review I have concluded in my presentation. Detection of drug resistant natural water bodies. not only resistant bacteria, but also antibiotic resistant bacteria, included gene resistant bacteria, are found in source water and also in treated drinking water. So the prevalence of antimicrobial multi-drug resistant E. coli are also found in drinking water sample from the Hyderabad. So the scope of my work is that as we know, everyone is water is the essential component of life. Its safety is important for good health. However, it could be the source of the dissemination of antibiotic resistant if it is not protect. Infection caused by antibiotic resistant bacteria is a challenge for the community and also public health. So that we design for this study, we focus on the determination of presence of antibiotic resistant in pathogenic bacteria from water sample. These are my two research objects. One of the to identify and quantify the pathogenic bacteria and to quantify antimicrobial resistant bacteria in bulk water from the Mehran University in Jamshoro. This is the methodology that we design. Very first, we collect the sample. We collect the sample from Advanced Water Center that is present in Mehran University, and I collect the sample in pilot scale lab. That water, tap water, and that tap water come from the main treatment plant of Mehran University. Then we transfer this water, physical and chemical parameter also done, and isolation of bacteria on different media. We selected 5 different bacteria: E. coli, Vibrio cholera, Salmonella, Shigella, and Pseudomonas. These 5 targeted bacteria, we take the CFU on specific media. I have showed the names on these media. And then again, we determination of antibiotic resistant bacteria by method using disk diffusion, and we check the zone of inhibition, and this experiment done on MH media. MH was Moller-Hinton media. This is the table of physico-chemical parameter: 32 temperature on that time water, 7.5 is normal pH, and 583.7 is TDS, and 898 is EC, 0.5 is salinity, and last is 16.8 is turbidity. And this result show the table of resistance. This table show the pattern of resistance of bacterial isolates of water sample of this study. As you can see, that the isolates are resistant to the majority of antibiotics. So everyone see the imipenem is the best drug, and like rifampicin, strepsin, bacitracin are not good drug. These bacteria are highly resistant most of the drugs. These antibiotic sensitivity test this diffusion method of pattern bacteria with the zone of inhibition. This is 8.2 millimeter, something like different points of dose, zone of inhibition present on media plate that were recorded, and I'm showing in this.
Sanam, you have 1 minute only.
Okay, I will conclude. 5 pathogenic bacteria, E. coli, Salmonella, Shigella, Pseudomonas, and Vibrio cholera were tested of the antibiotic resistant profile. These 5 bacteria are highly resistant against bacitracin, metroidazine, and ceforzane. Targeted bacteria were sensitive against imipenem, cefazine, and ciprofloxacin. All observed pathogens were found highly resistant. However, imipenem, cefazine, and these are. So in our study, we conclude that imipenem and ciprofloxacin are good drug of choice. My future research recommendation is this study could be extended further, each more antibiotic resistance of pathogenic bacteria against more drug of using molecular techniques. In vitro kinetic study experiment may be carried, which will give more appropriate dosage and time. Strict measures should be taken to reduce rational prescription of antibiotic. Awareness workshop and seminar should be arranged to improve the rational use of antibiotics. Thank you. This is the very
Yes,
serious
thank
issue.
you, Sanam, for very good presentation, and your work indeed is a very scientific one.
Thank
Do
you.
we have only one question?
Yeah,
Is there any question?
for
No,
me.
there is no question. Sanam, thank you
Okay.
very much. We move to next
Okay.
presenter.
Okay.
I
Thank you so
invite
much.
Mr. Nasibullah Khan Bazai, who is going to present water scarcity challenges in Balochistan, Pakistan. Over to Nasibullah Saab. Nasibullah, are you there? Nasibullah, are you there? I believe he is not here. So we'll move to next presenter. And I now invite Noorun Nisa Ghangrol and Aisha Tajammal for their presentation on Langnaria Cicraria, a possible remedy for emerging global challenge of superbugs. Over to Noorun Nisa or Aisha Bibi. Do we have the presenters? I believe they are not here. So we move further forward and I invite Arsalan Sheikh or Junaid Ahmed Kuri for their presentation on characterization and disinfection of biofilms formed on different pipe materials of
Hello.
drinking water distribution system.
Hello.
सर नूरुन निसा सर ज्वाइन कर चुकी हैं.
Nurinisa, okay.
Yes, sir. Is everyone watching my slide?
Yes, make it full screen. You have 10 minutes.
Okay, sir. Assalamualaikum to all of you. Here I am Nuranisa Ghangro, and my co-author is Aisha Tajamal. Here, the topic of our research paper is Legionnaria caesarea, a possible remedy for emerging global challenge for superbugs. Here are my table of contents, which I will go through it, and here is my abstract along with keywords. So, antibiotics are the chemicals produced by microorganisms and plants against other microorganisms, while capability of microbes to evolve and to prevent its effect is antibiotic resistance and considered as major cause of disease and death worldwide. The resistant strains of bacteria enter into water from human, animal, fecal, and industrial origin, which can ultimately spread these superbugs to water indigenous microorganisms. However, there are many different factors which microbes are acquiring resistance against antibiotics, which includes previously use of antibiotics, overprescription when patients do not finish the entire antibiotics, and so on. It also includes lack of hygiene and sanitation, unavailability of newly discovered antibiotics, and genetic mutations. We know that about 90% of the bacteria strains show resistivity against more than one antibiotic, and about 20% are resistant to at least 5. Pakistan has flora about 6,000 plant species which are being used as therapeutic means, but among them only 4 to 600 are being used in medicinal purpose. But still there is need of indigenous plants wealth that is unexplored. The development of multi-resistant superbugs and continuous appearance of superbugs with less vulnerability to antibiotics is elevating the threat to new battling techniques. The fresh plants were reported for great antimicrobial activity than the dried plants. Until now, several plants have been used against antibiotic resistant bacteria, including Pakistani spices like cumin, cinnamon, cardamom, and clove, and many other plants have been used against number of antibiotic resistant bacteria. Here is the profile of my plant which used, which includes its division, class, order, family, genus, species, group, duration, and its local name is Loki, we called in Urdu language, and common name is bottle gourd. And here is my problem statement that overconsumption of antibiotics and poor sanitation and hygiene aggravating current problem of superbugs and is continuously challenging to scientific community. Drinking water is the main vehicle to spread the enteric disease, and currently in Pakistan, many antibiotics have been reported due to the loss of their effectiveness against microorganisms. Hence, there is urgent need for novel and cost-effective drugs extracted from botanicals to counter drug-resistant microbes, as well as regulatory reforms so that clinical development programs become equitable, feasible, rigorous, and clinically relevant. This study was conducted in US Pakistan Center for Advanced Studies in Water, Mehran University Engineering Technology, Jamshoro. And the extract was prepared by taking locally plant as bottle guard was selected, and extract was prepared by using the water as a solvent. Water sample and isolation of desired microbes. It includes that water sample was collected from the same institute of washroom tap water, and the isolated microbes were clarithromycin resistant microbes, which were E. coli, Salmonella typhi, Shigella flexneri, and Staph aureus. After that, an antibiotic susceptibility test was performed using antibiotic disc as control and plants extract as treatment groups. All the test and control groups were subjected under proceedings using culture media having required microbial inoculum. All products were incubated at 37 degrees centigrade for a day. After incubation, the zones of inhibition were measured, and their values were standardized by using the National Committee for Clinical Laboratory Standard Institutes. Further, MIC tests were conducted to evaluate the plant activity against antibiotic resistant microorganisms. My results includes, here you can see the microbial inhibitory activity or bottle guard using the disk diffusion method, in which you can see that antimicrobial activity of crude extract of a bottle guard was investigated against clarithromycin resistant and sensitive pathogen via using the zone of growth inhibitions. At the concentration of 5 microliter per 50 ml, crude extract of bottle guard showed minimum and maximum activity against the clarithromycin resistant and sensitive strains of E. coli, that is 8 micrometer, and Shigella flexneri 8.6. In contrast, the clarithromycin resistant strains of Staph aureus was found to be inhibited significantly, that is 10 micrometer at the same dose. Upon testing the concentration of 10 microliter per 50 ml of the bottle called extract, we found minimum antibacterial activity against E. coli, whereas flexneri 9.3 millimeter and gram positive, that is Staph aureus, that is 14. In resistant strains, the only activity was observed against E. coli, that is 8 micrometer. The most promising results were there for disc with 30 microliter extract, producing measuring the mean along with their standard errors against E. coli, flexneri, Staph aureus, E. coli, and the highest one was, that is 16 micrometer. And the best activity you can see here, that against the concentration of 25 microliter, it showed the best resistance, that is 25.3 zone millimeter. Here are some the pictures. You can see that how much zones they showed during my research work. And after that, here you can see that antimicrobial activity of bottle guard extract against E. coli at 1600 nanometer optical density was taken to observe the growth for MIC. The highest activity of MIC was observed against gram positive then gram negative bacteria. Bottle guard showed the highest MIC at 500 microliter per ml against sensitive E. coli at 0 hour and 24 hour. The best activity against resistant at 50 concentration E. coli was observed at 500 microliter per 50 ml at 0 hour, and it showed the highest MIC at 250 microliter per ml at 24 hour concentration. 24 hour its time. Then it again became ineffective at the bacterial growth, and then inhibition was observed at 1 microliter per ml. Here you can see that effect of different doses of Legionnaria caesarea against the Salmonella typhi. It showed the highest MIC at 250 microliter per ml against sensitive Salmonella typhi at 0 hour, whereas at 125 microliter per ml, the best activity was observed at 24 hour. Against resistant 100 concentration Salmonella typhi, MIC was observed at 125 microliter per ml concentration at both 0 hour and 24 hour. Then at 0 hour it became ineffective, and then again it showed its activity 1 ml per ml of while at 24 hour the plant activity showed static results. Here you can see After that, here you have it showed against Shigella flexneri, in which the highest MIC was observed at 250 microliter per ml, and against the sensitive Shigella flexneri at 0 hour and 24 hour, all remaining doses were showing a static behavior, while against resistant 25 concentration Shigella flexneri, it showed the highest inhibition of 500 microliter per ml at 0 hour and 250 microliter per ml at 24 hour. Then it showed inhibition at 1 ml per ml. And here you can see against the Staph aureus, in which the MIC at 500 microliter per ml was observed against the sensitive Staph aureus at 0 hour and 125 microliter per ml at 24 hours. Against resistant 25 concentration, the Staph aureus highest MIC was observed at 500 microliter per ml at both 0 hour and 24 hour. It concluded that the suppressing activity of natural products or remedy was identified against antibiotic resistant bacteria. Clarithromycin resistant bacteria were isolated. Best colonies of E. coli were resistant at drug concentration of 50 microgram per ml. Staph aureus, Salmonella typhi at 100 microgram per ml, while Shigella flexneri and Staph aureus at 25 microgram per ml. Sensitive and resistant bacteria both were examined. The Legionnaria caesarearia, that is bottle gourd, showed best activity against the Staph aureus, but no activity was found.
Noor, your time is over.
For
Please try to finish it.
Yes, sensitive and resistant typhi. My future recommendations are that different drug-resistant bacteria must be isolated. That effect of these vegetables on different antibiotic-resistant bacteria and their synergetic effect must be further studied, and detailed studies on potential microbial agents must be followed. Here are my references. Further references. Thank you.
Thank you, Noor. Very well done and indeed very excellent presentation. Do we have one question?
Yes, sir.
Yes, sir. I have a question.
Okay, please go ahead.
No, this is very good presentation. I guess you have worked very well. So my question is, you have tested different bacteria, but you should have some positive control or something which is drug resistant or something, so that you can analyze that your strains, actually your plant is actually beneficial in the sense that even it can affect the drug resistant basic bacteria which are pre-known or something. So the positive control is missing, and these bacteria, you have mentioned, I guess these are separated from the water samples. So not all bacteria in the water samples are drug resistant or something. Maybe E. coli you have tested was previously it is already sensitive and you are testing drug on that. So what is the effect of that? It will so we should mention that a positive control first. And yeah.
Hello.
Yes.
Sir, sorry, your voice is missing during your question. Sir,
okay, no worries. We can take this question later on. Thank you, Noor, for a very excellent presentation. Thank you.
Thank you, sir.
So now we move to next presenter. I request and invite Mr. Arslan Sheikh and Junaid Ahmed for their presentation on characterization and disinfection of biofilms found on different pipe material of drinking water distribution system. Do we have Arslan Sheikh? Yes, please share your presentation.
Yes, sir.
Yes, please share your presentation. Yes, make it full and then start. You have 10 minutes.
Bismillahirrahmanirrahim. Hello, everyone. This is Arslan Sheikh from US Pakistan Center for Advanced Study in Water. Today, our research, I am going to represent characterization and disinfection of biofilm formed on different pipe material of drinking water distribution system. I'm research scholar at US because one of my colleague, Mr. Junaid Ahmed Khori, he's also helped me for this research. In this research, our supervised director, US Pakistan Center for Advanced Study in Water. Let's start our presentation. These are contents which will cover our presentation. As we introduction, now come to what's introduction. As we know all that every human being some basic need, but water is most essential, basic every human, including any type of age group. Safe drinking water is a big problem all over the world, especially developing countries, and where water scarcity is a great problem. Actually, water treatment system produce high quality drinking water, but unfortunately, however, the quality deteriorates as travel from the treatment plant towards end users. Microorganisms attached to the pipeline, biofilm formed on inner pipeline surface. Now to the biofilm. Biofilm is a collection of several types of pathogenic microorganisms, including bacteria, viruses, fungi, and protozoa. Fungi is one of the most common biofilm formed of eukaryotic organism into the water distribution as a result of clogging lines, deteriorate water quality, like bad and odor taste. These are causes of biofilm formation, persist the treatment facility, nutrient availability, rough surface, and corrosion. How to biofilm formation? First of all, free floating microorganism to attach in the inner surface of the pipeline. They are begin slimy extra polymeric substance to form colonies on inner surface of the pipeline. When biofilm is fully matured, to deattach in the water towards end users. Problem statement: presence of biofilm is one of the major issue in drinking water distribution system because it deteriorates water quality, taste, and odor, resulting adverse impact on human health. It causes chronic disease. Therefore, the identification and characterization of biofilm in drinking water distribution system is very important, will be beneficial for removing pathogenic microbes from water distribution pipelines. These are objects to design and fabricate biofilm forming reactor in the real drinking water distribution systems. US Pakistan Center for Advanced Study in Water, Jamshedpur. Number 2, to test the vulnerability of UPVC, PPR, GI pipe materials for biofilm formation. Number 3, to disinfect the formed biofilm on different pipe materials of drinking water distribution system with 1.5 milligram per liter hypochlorite disinfectants. To the methodology to design and fabricate biofilm forming reactor. This is model we have supposed to design different views how to install coppins in the pipeline. These are coppins. These are coppins dimension, area 2 inch, length of pipe 4 feet, diameter of the pipe 4 inch, control valve also available, floor measuring device, different pipe materials, PPR, galvanized iron, UPVC. After the installation of the biofilm reactor, biofilm was allowed to develop for 3 months. Biofilm reactor is shown in figure 1. Drinking water distribution system of Mehran University of Engineering Technology treatment plant location map is figure 2. First of all, installation of sampling device after 3 months biofilm formation, then biofilm extraction through CTEP method, then biofilm extraction, then DNA extraction through CTEP method, in sequencing, metagenomic sequencing analysis. Similarly, bulk water sampling, physical analysis, chemical analysis, DNA extraction, and metagenomic sequencing analysis. Bulk water sample will be collected from this distribution system. Biofilm sampling before and after biofilm formation. These show the before and after biofilm formation coppins. Shotgun metagenomic biofilm sample through then DNA extraction through CTEP method, metagenomic sequencing, library preparation, library pool, and metagenomic sequencing phylogeneticity. These are physical and chemical parameters of bulk water sample. These are same result to observe the changes in biofilm structure before and after disinfection. We analyze through same and observe change in structure of the biofilm. Same is scanning electron micro is a powerful technique for realizing the fine structure of system size and information localization has been applied to biofilm. It has also been of special importance in clarify biofilm structure for understanding the physiology and ecology of these microbes. These are live and dead cell results. Bacterial viability test, live and dead cell on fluorescent microscope, controlled biofilm without disinfectant dose. The result shows that the bacteria cells are alive after disinfection. 1.5 milligram per liter dosing. Continue 20 hours. The cells are dead. To check the effectiveness on disinfection on biofilm, we have used 1.5 milligram per liter hypochlorine disinfection. To check the effect of, we have used the fluorescent microscope from the cells, live and dead cell checks. Conclusion: a prototype reactor for biofilm formation was designed to install and operate in real drinking water distribution system at US because WMUT. Biofilm formation was monitored 3 months on different pipe material, including UPVC, PPR, and GI. Biofilms were disinfected by chlorine dosage of 1.5 milligram per liter for 20 hours. Biofilm reduction was observed at 1.5 milligram per liter chlorine dose in fluorescence microscope experiment and same analysis. This monitoring system could be successful system for monitoring biofilm contamination in the distribution network with efficiency for bacterial load reduction with the proper disinfection process. These are some references. Thank you.
Thank you very much, Aslan Saab, for making your presentation in time. Do we have only one question?
Sir, I have a question. Mr. Aslan.
Please introduce yourself.
My name is Asif. I belong to US Pakistan Center for Advanced Study in Water. So, Mr. Aslan, you just mentioned the sequencing analysis. I want to ask that which method you will follow the sequencing analysis?
Shotgun metagenomics.
Shotgun metagenomics. Okay. Any application?
Sorry?
Any application of shotgun metagenomics?
I don't understand your question. Please.
Maybe later on we can move to this question. Now, thank you very much, Arslan Saab, for your presentation. Now I invite the next presenter, Mr. Tahir Rafiq, who is going to present addressing fluoride fluorosis problem and its mitigation in the groundwater of third desert of Sindh province of Pakistan. Mr. Tahir Rafiq, sir. Do we have Tahir Rafiq? Or we move to next presenter? So next presenter is Miss Rubina Jaffrey, Senior General Manager from Hands, Pakistan. She is going to present Water for All, Food, Energy, Water Nexus. Do we have Miss Rubina?
I am here.
Yes. We can see you. Please share your presentation.
I have shared. Can you see?
No, not yet. Yes, now we can see your presentation. Make it full. You have Ms. Rubina Jaffrey, 10 minutes.
Yes, the presentation is I am Rubina from Rubina Jaffrey, working as a General Manager, Monitoring, Evaluation and Research Program in Hands. My presentation is Water for All. As you can see, I will discuss with the nexus of water, energy and food. So further I will move. We all know the water is essential for our life, and so we can discuss further about the nexus of the water. The food energy nexus is a speedy and very independent factors. In case of Pakistan, this relationship or nexus stands for very firmly. It is very deep binding and has far-reaching socio-economic dependence and consequences. So further, before going to the discuss the nexus of water related to food and energy, I will show the importance of the water and water treaties. It's very important to understand the water treaties and water issues. On this slide, I will discuss river flow from many countries and border. We all know, as in the past and even now, there has been always a rise in water-related dispute regarding the legal ownership of the rights of the water. And the first water treaty, which is reported in 2500 BC, कब्ल मसीह में ये पहली मर्तबा की बातें हुई थी, और but now There are nowadays currently we can see there are 276 trans-boundary river basin in 148 countries, including territory with 200 transboundary aquifer, and 90% of their territories within one or more transboundary river basin are in 39 countries. We can see the importance of the issue of the water, how this is creating the conflict and the treaties among the countries. But in Pakistan case, the next slide is showing that in Pakistan, river Indus is the largest river in Asia. And in Pakistan, Indus River formed the lifeline for bulk water supply through the network of dams, barrages, and irrigation channels. Similarly, water rights of nation have always been and remain today the flash point for water-related conflict. Water was reported, which was reported in to 1941 before partition among the between the Sindh and Punjab. So this situation was occurred in 1941. This dispute was settled. The dispute was settled in 1945, and through the commissions of Roy Commission, an agreement was termed with the name of Sindh Punjab Agreement. So the conflict and issues was resolved at that time. But after the partitions, we all know Indus River is a life sustaining between Pakistan and India. Since independence of 1947, flashpoint for water war between Pakistan and India has developed. So to resolve this issue, the Indus Water Treaty between Pakistan and India was signed in 1960. In view of the gravity of the problem in Ayub Khan period, who was the president of Pakistan. So after the water, so I will discuss before moving further the water requirement of Pakistan. There's crops like in Pakistan, most of the crops, Pakistan, we know that Pakistan is an agriculture country and more crops require more water. And like sugarcane and rice and cotton, they require more water than the other crops. Crops in Pakistan is independent on river Indus, independent on the river Indus. So mostly crops depend the water of the river. Indus, sugarcane and rice require more water. I have already discussed. Rice need constant irrigation and overall water requirement in 60 to 70 acre inches. However, the sugarcane required sugarcane required 96 acre inches. Similarly, we can see the trend of the cotton and rice tobacco. They are the most important crops in industrial crops. They contribute significant to the GDP, rural economy, labor absorption, and employment. So these crops have the very important role in the GDP. Demand and supply is directly proportional to the population increase. So demand is increasing as the population is growing. The burden of the providing food and water to these hungry arid soul become more resources and demand of the stepper. The person required about 100 liter of household purposes each day, but required between 3,000 and 500, 3,000 and 500 liter to produce his and her food. So we can understand the situations which is contributing to the water issue. It is also estimated that 36% of the groundwater is classified as highly saline. So this issue also facing in Pakistan. This slide is, we can present this slide, the water footprint of the crops in Pakistan in the comparison of the global scenario. The independence and relationship between food and water can be very well understand when we look into the cropping pattern of the country. Pakistan has the two crop seasons, Kharif and Rabi. So water requirement depend on the water requirement depend on the both crop seasons. Pakistan has the world fourth highest rate of the water use, and the water intensity rate, the amount of the water in cubic meter under per unit of GDP, it is the world highest. This suggests that no country economy is more water intense than the Pakistan, or category wise this slide also showing that Pakistan use more water than the global in comparison with the global scenario. How we can relate the water with the economy? Pakistan has the fourth highest rate of the water use. Its water intensity, it's also show the water intensity rate, the amount of water in cubic meters used per unit of the GDP is the world highest. This suggests that no country, I have already discussed this in the previous slide, the country water availability has reduced.
You have only 2 minutes left. Please try to finish.
Sorry.
You have only 2 minutes left.
So I have discussed on this slide how the economy of the Pakistan is depending on the use of the water, and this is also this slide is also showing the use of the cubic meter of the water required for production of the 1 ton, which is showing that sugarcane and poultry beef is using more water. Water stress level on this slide I can discuss is a declining factor, and 2025 we will be in the phase of absolute water scarcity, which is going down. The issue is very important for in the current scenario. Yes, Pakistan, which I have already discussed, they have Indus water treaty that Pakistan is depending on the water which is flowing from the India, or the flow of the water is reduced at 3.76 from 1991 to 2017 to 2018. So this is also this graph is showing the fluctuation of water, fluctuation of the water flow due to the conflict of the India. I will, at the end, I will discuss the nexus of the electricity, water nexus with the energy, and which is depend for the development of Pakistan. Cheap electricity is very crucial. It's one most important factor for the sustainable industrial growth, expanding employment, improving health and education. And it's also contributing the Human Development Index in Pakistan. The hydro-based energy output potential for the country has been calculated. But,
Madam, your time is over. Try to conclude.
Okay. I will come on the conclusions that I have discussed. The water flow is depend on the flow from the India. So water scarcity and water flow is not consistent. Again, some crops require more water and depend on the on that crops like sugarcane and major crops of the water being consumed by the two crops, especially crops rice and sugarcane. The technology is not updating and farmers not using the traditional farmers using traditional method. So need to update the technological aspects and into introducing the new techniques to establish the to develop the nexus of the nexus with this context. Pakistan is consuming more water and agriculture and crops. Similarly, shortage of energy is also due to the shortage of the electricity and shortage of the reservoir, which is not enough infrastructure and infrastructure of the dams and canals and other these systems is very poor, or need to develop this structure and these structures. So this was the concluding page, and thank you very much. I have concluded overall. Thank you very much.
Thank you very much, madam. Thank you very much. Now we move to our last presentation of today. I request Mr. Sher Mohammad and Parvez Khalid from Institute of Geology, University of Punjab, Lahore, for their presentation on physico-chemical analysis of groundwater extracted from late Quaternary aquifer of in southern Peshawar Basin, Pakistan. Over to Sher Mohammad Sahab.
Yes. Bismillahirrahmanirrahim. The topic of my research is physiochemical analysis of the groundwater, which was extracted from the district Nusheira, which was my research area. Here, these are the outlines on which I will try to discuss. First of all, I would like to tell you why the water is so significant for life, and what is the significance of the constant supply of the water. Without constant supply of the water, can we survive? Or without proper management of water resources, how we can survive? Okay, first I am going to discuss why water is so significant in life. There is very simple sentence: there is no life without life. There is no life without water, because water is a basic constituent of the all living organism. Water has such chemical properties, that is why it is known as the universal solvent. It can solve maximum number of other compounds within itself, which is there is not any alternative of this chemical compound. Water is responsible for biosphere, with the exception of this, water also plays a basic role in hydrological cycle. Water is a unique chemical compound which exists in three different forms: that is solid, liquid, and gas. In solids, ice, snow; liquid, snow, water, sulfur water, and etc. and so on. Now, second thing, how water supports the life. It is a basic component of the earth. Now,
Yeah,
in
it's okay.
these slides, I would like to. I have given some brief data about why water is significant for the life. Nearly 71% area of the surface is covered with water, and there is only 29% area which is consists of lands, mountains, countries, and islands, and etc. But this 71% area which contains the water, out of this only 97.2% water is saline water, which is not usable. In different literature, this percentage is different. In somewhere it is 96.5, somewhere 97%, sometimes 97.5%, etc. So among the remaining 29% area, we have to find out the fresh water, which is a basic need for all living organisms. Here, a brief chart shown in which 97.2% water is shown by a big circle, which is a saline water. Among saline water, remaining 2.8% water, 2.1% water is in the form of the glaciers, which is not usable, and only nearly 1% water is fresh water. Now, now in the year of 1995, World Bank Vice President of World Bank declared that in the next century, the war will be fought on the basis of the water sources. In 2016, United Nations Environment Program claimed that up to the year of 2030, half of the world body will have to face severe stress of the water. In 2017, UN Secretary General Ban Ki-moon claimed that up to the year of 2030, the water will face a 40% shortage of the water. According to the UN report, globally 3 billion people are facing the shortage of the water. With the exception of this, the World Resources Institute also claimed that up to the year of the 2040, 17 countries will be included in the list of which face extremely high baseline water stress. Among these 17 countries, 12 countries are from Middle East and North Africa. Due to this shortage of water, their economy may reduce from 6 to 14% of their GDP up to the year of 2020. This is the list of 17 countries which have to face an extremely high baseline water shortage. And in this list, Qatar is first list at first number, Israel is at the second position, and so on. There is also in the now let me show. This is also a map here. Pakistan, the color. Let us focus on the color of the Pakistan. That is red color. This color shows these are those countries which have to face extremely shortage of water up to the year of the 2040. It means that we must be careful for the saving of the water. Lekin saving of the water, which is our basic need. Now Pakistan is agriculture country, so we must be careful regarding the water resources, and basically we have to focus on the water management. Up to the year of 2025, Pakistan has to face severe condition of the water shortage. By 2040, Pakistan is predicted to be one of the most water stressed country in the South Asian region, while at the same time it will be the 23rd most water stressed country in the world. It is according to the report of WRI. Almost 30 million people in the Pakistan don't have any access to the clean water. Pakistan is an agricultural country, so we need constant supply of the water. Agriculture pay 21% of our total GDP and 40% of the labor force. Livestock, which pays 9.7% to our GDP, it also needs to continue supply of the water. Due to the shortage of water, we have to face different problems. If there is less water, there will be less production of the crops. If production of the crops is less, then there will be less input material for the industry, so industry will collapse. When industry collapse, there will be loss of the job, so economy will decline, unemployment will rise, and so on. Currently Pakistan is at the fourth position of among those countries which are using water more highest rate. At the highest rate.
Sher Saab, you have only 1 minute to go. Please try to
okay,
try to conclude.
Anyway, okay, let me go to my conclusion. This is my study area, which was in Nowshera, that is part of the Nowshera Peshawar Basin. Now I am going to conclude. This is a flowchart for water contamination table. This is again water. Let me move towards these. I have collected the total 12 number of samples. For those I have done physiochemical analysis. These are the WHO standard and standard NSDWQ Pakistan with reference to Pakistan. So basically, in this table, I have given the data collected after conducting physical analysis. From this data, I observed that and what is the need of monitoring the pH value limits are from 6.5 to 8.5. The most of the microphone samples exist within this limit of the WHO. Only one sample, that is S188, has a little bit high value. Who are facing water shortage. Well, temperature is of all the water sample was within the defined limit of the water is increasing, but the water this is shown the calculated values of TDS, total dissolved solids, and the electrical conductivity. In this case, most of the TDS value for the most of the sample is below the prescribed value, but this value is not too below that they can be disturb the water. Okay, let's move. If the TDS value is much low, let's suppose if TDS value less than 100, then water will be suitable, but it may cause hair fall and heart attack. Global environmental change. This is TDS related electrical conductivity source. Let me move towards the chemical analysis. I have calculated the percentage of these chemical components: calcium, magnesium, sodium, and after calculation, the result obtains are these are given by. After these results are represented graphically, and then when I can compare these results with the standard values which are defined by the WHO, these results are these chemicals. The values of these analyses are less than the limited value of the announced by the WHO. So these samples of water are okay and they can be used. This is a carbonate, bicarbonate, chlorine, and sulfate. Okay. It is the same thing which I have mentioned. Physicochemical analysis. Okay. These are the same thing which I have described. Thank you, sir.
Thank you very much, Sher Mohammad Saab.
Thank you, sir.
That concludes our presentation. So at the end, we may take one or two questions regarding all the presentation. If anyone still have questions, he or she can raise the hand. First, Barira Fatma.
I just have a question from Sanam and Noor. They have in their research they have presented. Can you hear me, sir?
Yes.
They have presented that they have conducted उन्होंने just एक simple ये research की है कि बहुत अच्छी research है कि antibiotic resistant find out किया है उन्होंने drink water के अंदर, लेकिन उन्होंने अपनी टेस्ट के अंदर सैंपल के अंदर ड्रिंकिंग वाटर यूज किया है. तो इग्नोर माय इग्नोरेंस और माय लैक ऑफ नॉलेज के मेरा तो ख्याल है कि जो एंटीबायोटिक रेजिस्टेंट बैक्टीरिया होते हैं दे आर सपोज टू बी प्रेजेंट इन वेस्ट वाटर. सो ड्रिंकिंग वाटर को यूज करने का क्या पर्पस है फॉर दैट टेस्टिंग?
Sir, may I answer that question on behalf of Sanam?
Sure. Anyone can answer.
Okay. So basically, there are many literature, present in which you can see that the wastewater is being included in the surface water. For example, River Indus, there are many papers and resources where you can find out that wastewater is being included in this surface water. This is the reason that the drinking water which we are, in Karachi and Pakistan is being supplied, or any other city which is connected to every city is connected to the river industry actually. So basically there is said that there are industries and different villages beside this river Indus which are polluting the river Indus water. That's why these bacteria are coming in the surface water or drinking water, and other sources are household water. In the tap water. So other sources are households and hospitals which dump their water directly into the water sources, and and Hyderabad specifically, there are the lines which are wastewater lines and drinking water lines. They are parallel to each other. Once drinking water is closed and these wastewater lines get leaked, this water comes again into the drinking water sources, and these bacteria can spread to the end user, and they can cause diseases and so on. So I guess I have answered your question.
Thank you, Junaid Saab. Do we have one more question?
I have a question. My
Please
name is
introduce
Dr.
yourself.
Ashila.
Yes.
I'm a manager at Hands. I'm also a dentist. So I wanted to know, while collecting the sample, this question is for both the speakers who talked about the disk diffusion method and the resistance and sensitivity towards the antibiotics. So how was the cross-contamination prevented during the processing of the specimen? Not just the processing, at the time of the collection of the sample, how the cross-contamination was prevented. Because, through cross-contamination, the bacteria might have entered into the sample. So there must be a method. I just wanted to know what did the PI do to sort this problem?
Okay, let me answer that question. So, whenever anybody works on the water samples specifically, so he needs to be, to be careful for some things. Like when he's collecting the samples, so he should be wearing the gloves. He or she should be wearing gloves and PPEs. And other thing is the collecting material, the material in which he is collecting the sample, it should be sterile and
Sterile, yeah.
pure. Yeah. So we have collected the samples and we have processed it, so we have cared everything such that no external contamination could include in the water samples. So we have handled in that, and all the samples were processed in the sterile conditions in the laminar hoods, and ethanol sprays and uh, disinfection through detergents was also
And the
carried out.
sterilization of the equipment.
Yeah, yeah.
Thank you, Junaid Saab, and thank you all the participants. That's conclude our session for today. I want to thank Mr. Fareed Alam Saab for a very nice presentation, a thematic presentation on water, food, energy, Nexus. And I also want to congratulate all the presenters who presented today. Your research was very new to us, and it's very fascinating to see all the young students doing very good research in the universities. And I also want to thank Kashif Zahib sir from RIFA for providing the IT facilities. Thank you and goodbye.